Thank you for the introduction, and thanks very much to the organisers for having me here. I'm going to give you a very brief talk about the role of metals in Alzheimer's disease. I'm going to focus mostly on iron in this presentation, and also, because of the nature of the meeting, speak about the role of iron in aging itself. These are my disclosures. So just the 101 of this subject matter. Cortical tissue contains very high concentrations of endogenous iron, zinc, and copper. So the transition metals are used for many functional neurochemical events. And the metals are kept under strict homeostatic control, and the blood-brain barrier is generally impermeable to the movement of metals into the brain from the plasma. Now, there are many novel players in metal homeostasis that are implicated in Alzheimer's disease. And recently, the presenilins, the amyloid protein precursor, and tau have been described to participate in metal homeostasis. And today, I'll show you some of the data recently published about APP and tau and their role in regulating iron trafficking. In Alzheimer's disease, there's a marked dysregulation of metal ions, and this contributes to oxidation. Particular copper and iron are involved in redox chemistry. And this dysregulation of metal ions lies both upstream of the proteinopathy and downstream of the proteinopathy. This graph shows you data that we published over a decade ago, which simply measures the concentrations of metal ions in the brains of mice as they age. And compared to the baseline, you can see that shortly after reproductive life, there is a very marked increase in the concentrations of iron and copper within the brain. These are all relative to the baseline, expressed as percentages. In terms of absolute abundance, iron is by far the most abundant of the metal ions. And subsequent work has shown that the brain has a curious problem regarding its iron metabolism, and that is that if you trace the influx of iron into the brain from the diet and compare it to other tissues, data shows that it accumulates within the brain in a manner that is unlike other tissues. So the efflux rate of iron into the brain from the after entering into the brain from the diet is the slowest of any of the organs that are measured. And the half-life of dietary iron in the brain is the greatest of any of the organs that are measured. And so this is some of the evidence that shows that the brain has a tendency to accumulate iron with aging. Now, the accumulation of iron with aging has actually been described in all kingdoms of life. Here, for example, is a paper published in 1995 from Trevor Williams' lab that shows you the accumulation of iron in the body of aging Drosophila. And this event is something that we were very curious about because of the association with Alzheimer's disease that I'll go into in a minute. Indeed, other groups have contributed and shown that in primates, you will also see that there is an accumulation of brain iron, which has a functional consequence. And this very intriguing study published in 2010 from Sterling Johnson's lab, it shows that caloric restriction not only improves motor performance, but seems to do so by the virtue of decreasing brain iron in rhesus monkeys. And it has been also argued that in humans, there is an association between hippocampal iron concentration and the atrophy of the hippocampus and changes in memory that are associated. So we've drilled down into this chemistry in greater depth in C. elegans. I want to show you some new data which has just been submitted. And this is work of Gavin McCall in my lab. And what we noted is that as the C. elegans reaches the age where it begins to die off, so after the age of 10 days, there is a conspicuous rise in iron in its total body. So this is just taking C. elegans and measuring the content of iron per worm. You see that at 12 days, there's roughly a doubling of the content of iron within the worm. This doesn't happen in the long-lived DAF2 mutants at the same age, but introducing the DAF16 mutation, which abolishes the longevity, restores this elevation of iron within the worm. And this occurs concomitantly with an elevation in reactive oxygen species generation, which was measured by DCF at the same ages. So the argument is that the iron is the origin of the reactive oxygen species. And we can use immunohistochemistry to actually localize this iron. You can see that in a 12-day-old worm, which is an age where these worms begin to die off, you see accumulation of iron stained with the Pearl stain, and particularly within the nuclei of the gastrointestinal epithelium. And these are the nuclei which die off and are believed to be a major contributor to the actual event that leads to the death of the worm. So this curious elevation of iron that occurs in all organisms and in C. elegans allowed us to actually analyze it by chromatography. And we use size exclusion chromatography to define what is going on. So in terms of the total elevation of iron, we could separate out three major chromatographic peaks from C. elegans as they aged from 4 days to 13 days. And you can see that the peaks do change with aging. I'll show a graph of each of those three peaks to show what the changes are. So over from 4 days to 12 days of age, peak 1 increases. I'll use this pointer. Peak 1 increases significantly, peak 2 decreases significantly, and peak 3 increases significantly. Peak 2 we identified as being ferritin, and ferritin is a safe storage protein and is not expected to be able to hold the iron in a redox competent manner, could not be the source of the reactive oxygen species that are generated. So the culprits in terms of reactive oxygen species that rise in tandem with the elevation of iron, we argue therefore must lie in either peak 1 or peak 3. So most importantly, you can intervene with this. And obviously, we tried iron chelators of various kinds to try to impact upon this and see what happens to longevity. The one that we had the greatest success with is SIH, which is a compound that Des Richardson introduced into the literature, and it's been used quite a bit in various studies. And it significantly increased the lifespan of the C. elegans by 75% at maximum. And it appeared to target peak 3, not peak 1 or peak 2. Well, peak 2 contained ferritin, could not be the culprit, but peak 3 we believe is the culprit that's responsible for the effects that are mediated by the elevation of iron that leads to the shortening of the lifespan of the worm. So in summary, what we're saying is that the escape of iron from ferritin, that is the peak 2 that is lost with aging, lends itself to a form of iron that contributes to reactive oxygen species and is tractable to chelation with compounds like SIH. It's a low molecular weight iron species. So the significance of this is also whether it's relevant, obviously, to human disease, whether we can see it in neurodegeneration. This is where we're heading with this project. As I mentioned earlier, it's known that iron increases in the brain with age, and an excess of iron is involved with neuronal damage. This actually is established because primary iron lesions caused by genetic mutation cause dementia and Parkinsonism. And these include neurodegeneration with brain iron accumulation caused by PANC2 mutation, neuroferritinopathy, aceruloplasminemia, and Friedreich's ataxia. So it establishes that the accumulation of iron in certain brain compartments will indeed cause neurodegeneration. The question is whether or not this is also true of Alzheimer's disease and, say, Parkinson's disease. And indeed, there's a considerable literature that shows that there's an elevation of cortical and hippocampal iron in Alzheimer's disease, an elevation of nigral iron in Parkinson's disease, as well as all Parkinson's disease models. The Parkinson's disease models are actually very interesting in terms of the robustness of this literature. This is an X-ray fluorescence image of the increase of iron in the substantia nigra that occurs as the nigral neurons die. And the argument has been that the iron will react in a reducing environment with oxygen and generate reactive oxygen species. And this is where we think that the SIH might be actually targeting the event in worms. But what of Alzheimer's disease? Well, the literature is now rapidly filling up with instances where iron has been noted to be elevated in the pathology of Alzheimer's disease. And this in particular seems to be robust using MRI technology. And there have been a couple of excellent papers published recently by a group in Leiden in Holland, run by Van de Grond, who have shown by MRI signals that you get the elevation of iron, which occurs in tandem with cognitive changes in Alzheimer's disease. And in fact, it can differentiate early onset Alzheimer cases from late onset Alzheimer cases by this iron signal, which is significantly greater in the cortex of cases who come down with Alzheimer's disease at a younger age. We've spent a lot of work on beta amyloid and its interaction with these transition metals, and some of the drugs that have emerged from this work, drug candidates that have emerged from this work, have targeted the reaction of beta amyloid with extracellular zinc. And the principle of this is, as people get older, the ability to traffic transition metals within the brain fatigues. Zinc is released during synaptic transmission and tends to pool extracellularly. But at the same time, it has been known that the iron that's accumulating within the neocortex in Alzheimer's disease is actually partitioned into the tangle-bearing neurons. And this was first identified by Mark Smith and George Perry and has subsequently been corroborated. So for the rest of this talk, I'm going to focus on this neuronal iron as a relevant target. And the first new protein that I want to talk about in this regard is the amyloid protein precursor, which we are very confident facilitates iron export, and this, we propose, is the major function of the protein. This is some of the evidence of it, and that is that primary neurons that are incubated with radioactive iron and then washed, and you watch the efflux into the tissue culture media, you can see that the efflux from the APP knockout primary neurons is much less than that of the wild-type neurons. APP knockout neurons are more vulnerable to iron toxicity if iron is introduced into the media. But most importantly, APP interacts with the obligate iron export channel ferroportin. There is no other way for iron to get out of any cell. And APP, like the positive control ceruloplasmin, interacts with the ferroportin in pull-down assays from both mouse and human brain tissue and accounts for about 75% of the APP in the brain. In other words, by doing housekeeping, by doing bookkeeping analysis of this pull-down interaction, we believe that 75% of the APP in the brain is interacting with ferroportin. So it's a major interaction that we're talking about and has been corroborated recently by Dan Kosman's lab. When an APP knockout mice therefore accumulate iron in various organs, they accumulate iron within the neurons in the cortex, and also importantly, when challenged with iron in their diet, they cannot stop that iron from entering into the brain. Unlike wild-type mice where there's no elevation of iron in the brain after 7 days of iron feeding, the APP knockout mice does experience a significant increase in brain iron in both in the brain and also within other organs such as the liver. So the second player in iron homeostasis in neurons, we believe, is tau. And I want to make this point, which is an important one in the wake of the previous speakers, is that, of course, tau forms this dramatic proteopathy, but at the same time, the concentrations of soluble tau in affected tissue decrease. Now, here's a series of cases of nigral tissue taken from cases of age-matched controls and Parkinson's disease, which we recently published in Nature Medicine, that show that if you fractionate the tissue into soluble and insoluble fractions, the concentrations of tau in the tissue decrease as they rise in the pellet phase. Here you see concomitantly with the drop in tau in this series of cases, there is a significant elevation of iron in this affected tissue. And we did a larger series which corroborates that there is this drop in soluble tau. This, in fact, has been observed previously, both in Alzheimer's disease and in other tauopathies. So it's not really a controversial point. It's just a point in terms of what is its relevance to pathogenesis. So our argument is, could there be a loss in function? And therefore, what is the function of tau? Well, we recently published in that same paper that tau participates in neuronal iron efflux, and here we again see the iron efflux experiment being done, this time using tau knockout primary neurons. The red is the tau knockout primary neurons, and the black is the wild-type neurons, showing that the efflux is suppressed in the absence of tau. And tau primary neurons in culture accumulate iron compared to wild types, and this can be abolished by treatment with a chelator, and one of our favorite chelators that we've used over the years is clyoquinol. So to summarize a lot of work which is in this paper, the mechanism for how tau does this is by trafficking APP to the surface of the neuron, where it interacts with ferroportin and facilitates the efflux of iron 2 through the ferroportin channel and to be loaded into transferrin for removal from the cell. So without either APP or tau being normally functionally available, you'll get an accumulation of iron within the neuron, which can be toxic. And indeed, the tau knockout mice develop a dementia with Parkinsonism phenotype. Here is an MRI of the animal's brains done at different levels. We can see a characteristic wild type versus knockout. And you can see the very conspicuous ventricular enlargement, and what is less conspicuous there is the cortical atrophy, which we could also measure and was significant. So the brain shrinks in these animals by 12 months of age. And this atrophy is completely rescued by treatment with clyoquinol, the iron chelator I mentioned earlier. We have a paper in press which corroborates this. There was some debate about whether or not this was a reproducible finding. In our hands, it's highly reproducible, but it is strain dependent. And we compared two background strains in terms of tau knockouts for the impact in terms of Parkinsonism and cognitive loss, Black 6 and Black 6129 SV. And we could see that there was an impairment in the pole test in both strains, which was rescued by levodopa. So this is therefore Parkinsonism. And we could also see that there is an impairment of Y-maze performance, a memory test, which is confined to the original strain that we studied, but not the Black 6 strain. So there are some nuances in terms of the impact of the phenotype, which could be strain dependent. We're also aware that both diet and environment could impact upon this phenotype. But it is a robust change, and the neurodegeneration is very clear. So most importantly, we can rescue this phenotype by treatment with clyoquinol in both the pole test, and here you can see the increased time it takes for the tau knockout mouse to turn on the pole from 7 months of age to 12 months of age, which is abolished by treatment with clyoquinol, which suppresses the rise in iron. And similarly, we could rescue the deficit on the Y-maze, which we observed by treatment with clyoquinol. So here we're rescuing these neurodegenerative phenotypes by treatment with an iron chelator. This principle is actually being picked up in a important recent phase 2 clinical trial, which was published by a French group led by David Deveau. And this is one of the figures from that paper that has recently been published, which shows that treatment with the iron chelator deferiprone not only significantly dropped the iron signal that could be appreciated by MRI and the substantia nigra of the treated patients, but also this occurred concomitantly with reducing the rate of functional decline in the treated patients. This was a delayed start treatment protocol. I won't go into the complexities for the sake of time, but we feel it was proof of principle that treatment with a brain-penetrable iron chelator could actually be disease modifying for Parkinson's disease. And by implication, therefore, potentially for other tauopathies where there is an increase of iron, such as Alzheimer's disease. And I'll leave off with what the literature might say about this. These are the only two agents with the potential to lower iron that have been used in clinical trials in Alzheimer's disease. They're both small phase 2 clinical trials that reported some signal of efficacy, but potentially the way is open now to investigate this with greater rigor, and hopefully there'll be newer agents such as deferoprone that can be tested in more phase 2 clinical trials and see what the impact is on outcomes for cognition as well. So with that, I thank the group who've done all the work. I particularly featured Gavin McCall's work today on worms, and my collaborators at the University of Melbourne and at the Mass General Hospital. So thank you very much for your attention.